Welcome to the ORS Virtual Scientific Session, ORS Spine Section Virtual Research Update Series, Spine Biology, organized by John Martin and Sarah Goldbrand of the ORS Spine Section. I am pleased to introduce today's session organizers, Drs. John Martin and Sarah Goldbrand. John received his PhD at the University of Pennsylvania in 2015 and is now a postdoctoral researcher in orthopedic surgery at Duke University. His work broadly focuses on connecting spine biomechanics and spine health and is currently funded by an NIH K-99 Pathway to Independence grant. John also currently serves as membership committee co-chair for the ORS Spine Section. Sarah received her PhD from Rensselaer Polytechnic Institute in 2014 and is currently a career development awardee at the Philadelphia VA Medical Center, as well as research faculty in orthopedic surgery at the University of Pennsylvania. Her research interests include the structure-function relationships of spinal tissues, advanced imaging techniques, and tissue engineering. Together with John, Sarah serves as membership committee co-chair for the ORS Spine Section. At this time, I'm pleased to present Dr. John Martin.
All right, thank you so much, Shari, for getting us started, and thanks to everyone who's joining us online here. So like Shari said, today's session is organized by the ORS Spine Section. I'm going to turn it over to Sarah now to introduce our speakers.
Thanks, John. So our first talk today will be given by Professor Christine Lemaitre. Christine is Professor of Cell Biology and Tissue Regeneration at Sheffield Hallam University in the UK. She has worked on the intervertebral disc for the last 20 years and currently leads the Tissue Regeneration and Biomechanics Research Group in the Biomolecular Sciences Research Group, and is also head of research degrees for the College of Health, Wellbeing, and Life Sciences. Her research to date has led to over £20 million in funding, 80 publications, and a current H-index of 36, an I-10 index of 59, with more than 6,250 citations on her work to date. Christine, please take it away.
So thank you very much for the opportunity to share some of the work that we've been doing, looking at trying to develop a consensus methodology for intervertebral disc cell culture. And I'm presenting this on behalf of a very large consortia where we've been working together to try and develop this consensus methodology to pull together all the methods that everyone's using for cell culture. Now, the use of cell culture, sorry, I've got no disclosures. So the use of intervertebral cell culture is used in a number of different fields across the spine field. So we utilize in vitro culture systems for investigating basic pathogenesis, so where we can grow cells from non-degenerate or degenerate discs from humans or from animal models and grow those in culture and then manipulate various conditions to actually understand whether those things are linked to the pathogenesis of the disease. And that has helped us get a great understanding of what goes on during disc degeneration. In addition to this, cell culture is used quite a lot in the development studies, so where we want to understand where the disc comes from, what cells differentiate into disc cells, what are disc cells. These are all really important questions within the spine field. And then, of course, we can also utilize in vitro culture for mechanistic studies, where we want to try and probe the mechanisms by which cells interact with their environment or with the tissues, etc. And of course, then there's also the development of new therapies. And this can be gene therapies, cell therapies, biomaterials, growth factors, etc. There's a whole host. And people will use in vitro cell culture for all of these different mechanisms. So what we wanted to do was, first of all, to see whether there is differences across the world on the current methods that people use for isolating, expanding, and also culturing intervertebral disc tissue cells and also as organs or isolated cells. So what we did was we sent out via the ORS Spine Section a survey to actually ask people who do in vitro cell culture to submit their methods on extraction, expansion, and tissue and organ culture. And these were some of the different areas that we were asking about. And we were absolutely amazed by the response. So we had 36 different institutions who responded to the call. And this was absolutely outstanding and kind of blew my mind a bit in terms of how many people were really engaged in this important endeavour. And we've been working together now for about the past 6 to 8 months, where we've been trying to pull together what methods everyone is currently doing and then utilising this knowledge to develop the new methods going forward. So what I'm going to do today is basically show some of the data that we've pulled where we're comparing the different methods that everyone's submitted. So the first aspect is how we're extracting those cells. And actually, although on the survey we asked for any intervertebral disc cells, the majority of the methods that were submitted were talking about nucleus pulposus cells. And although some were looking at annulus fibrosus, what we decided first of all was, because the methodology for MP and AF cells can be different, we were going to focus first of all on the nucleus pulposus cells, and that's what I'm going to talk about today. So what you'll see within these graphs, and I've got a number of different types of graphs which I'm going to demonstrate, but what we've done is separate out the species that we're showing into the small animal models, which retain those large vacuolated notochordal-like cells, the larger animals, which again retain these cell types, and then the larger animals which lose them, very similar to the humans, and then of course the humans. So when we were looking at the actual extraction methods, there was a varied response in terms of which groups utilise pronase as a pre-treatment prior to enzyme treatments, such as collagenase, etc. But although most groups did use this. Now, there is actually an interesting paper that was published a few years ago who were looking at the use of pronase and whether it was required. And that actually demonstrated that in the more mature tissues, such as human and bovine tissues, pronase is not required, but in the less mature tissues, or those which have a very rich glycosaminoglycan content within the disc, that actually pronase is required. So this was quite interesting, and I'll come back to this later when we're talking about developing our methodology. Now, as well as whether people use the pronase, of those that did use it, what you'll see is there's a whole range of concentrations of pronase that were utilised within the different groups. Not as much as we saw, though, with the collagenase. So if you actually look at the different types of collagenase or other enzymes that are subsequently used to digest the tissue, you can see there's a whole host of different types of enzyme which people use to isolate the cells from the disc. The most common was type 2 collagenase, particularly in the larger animals, although in the smaller animals, it was type P collagenase. As well as the type, the concentration varied substantially, and also the time period that different groups utilised, although this was somewhat in relation to the size of the tissue and the maturity of the tissue, with the larger animals generally having longer duration of digestion than the shorter, more jelly-like tissues, which you would imagine can be digested in much shorter times. In addition to this, we looked at whether groups utilise cell strainers to isolate the cells following the extraction. And again, there was a mixed use of this, with more groups within the mouse, rat and rabbit not using them, but more of the larger animals do utilise them. Again, the cell size and cell strainer size, which was utilised, varied substantially, although the most common was the 70 micron cell strainer size. So as you can see, there's large variation in the current methodology that people are using, even within the same species, across the world within the spine community. And this demonstrates there is a need to try and pull together methodology, because if you use different collagenase or different time periods, and that might change the phenotype of those cells, or you may get more or less cells being extracted. So we need to generate some standardized methodology for the extraction methods. So if we now look at the expansion methods that people were utilizing to culture the nucleus pulposus cells, you also see a large variation. So I think the basal media that people use, although there is more groups use sort of DMEM or DMEM HAMS-F12, there is actually a variation of different medias used. Most groups use 10% fetal calf serum, although some do use more, and very few groups don't use any. Even down to the use of whether pyruvate is included in the culture medium, and actually some groups don't really know whether it's used, which is a little bit worrying. But most people knew whether they were using it or not, but actually this took a bit of probing, and this wasn't actually submitted to start with on the survey because people assumed it was already in the media, So media comes with this already included, but actually it's become apparent through the discussions that the pyruvate inclusion is actually an important feature that we need to investigate whether or not we need it. And again, the concentration does vary across the groups. If we look at something which I always thought was quite simple in terms of the inclusion of ascorbic acid, which is required for collagen synthesis and deposition. But actually, if you look, there is a large variation in the groups, whether they utilize ascorbic acid, again, what type of ascorbic acid they use. So this form, the L-ascorbic acid, is a short-lived ascorbic acid that really you need to replace regularly. You need to freshly replace it, which most groups do. But also the concentration of ascorbic acid which utilised varies substantially between the different groups. When we go to look at sort of whether people try and mimic the physiological environment of the disc, you can see that actually the majority of people are using kind of standard high glucose concentrations. Sorry, my legends got lost there. So the blue is high glucose, the red is low glucose, and the black was don't know. The growth factors generally are not used, and the oxygen concentration in the majority of cases is just standard culture conditions, which varies between 17 to 21% oxygen, depending on the altitude of the lab and also the humidity of the environment. So this is interesting because actually the majority of groups do not use physiological oxygen concentrations, which is more like less than 5, and the majority do not use the low glucose media, which again is more physiological. However, this is probably because if you try and grow out cells within low glucose and low oxygen, they often don't proliferate as rapidly. So normally the expansion is being used so you can have enough cells to do your experiments. So you want the cells to grow rapidly. When we then look at MP tissue and organ culture, so this is where you're keeping the tissue together either as a nucleus pulposus tissue explant or the whole organ system, we again see great variation in the levels and the concentrations of these different components. So the basal media again varies, although there is a greater number of labs which utilise the DMEM. And the fetal calf serum now is a bit more physiological, with more labs utilising lower concentrations or no fetal calf serum, and fewer labs utilising the standard 10% fetal calf serum we saw when we were expanding cells. The pyruvate is very similar to that reported for the cell cultures. If we look at the ascorbic acid, once again, there is quite a lot of variation in terms of whether ascorbic acid is utilised, which form of ascorbic acid, whether it's added freshly, and the concentration was less varied, although there is less groups, of course, doing the explants and the tissue organ cultures. With the growth factors, again, very few labs were utilising growth factors within these cultures. Glucose concentrations was, again, still more of the higher glucose concentration, although if you think physiologically, if you're now culturing a whole organ, then actually in the central region of that disc, it probably is more physiological because the glucose has got to diffuse through the tissue. Whilst if you're utilising explant tissues, you may want to use a lower concentration of glucose to mimic physiology. And again, oxygen concentrations were predominantly, apart from in the human samples, were predominantly the standard culture conditions. So from this data, we demonstrated that across the world, there is great variation in the culture methods that are utilised for disc cells and disc tissues and organs. And this demonstrated that we need to try and develop a consensus methodology, which is why it was so fantastic that we had so many groups that were engaged in this endeavour. So what we've decided is what we're working towards is 2 key papers, which are looking at the cell culture consensus methodology. Now, through the various Zoom calls, etc., that we've had many discussions of how far we go with this. Do we actually go all the way to trying to mimic the in vivo environment and controlling things like glucose and oxygen, etc.? And actually what we've decided is, first of all, we need to get a method straight for the simple components first, and then we can go on to the more complex modelling of the in vivo environment, etc. So what we're doing at the moment is we're looking at developing consensus methods for the NP cell extraction, and we've got small groups working on species-specific methodology where we've grouped together those same groups that I showed in the graphs. So the small animals with notochordal-like cells, the larger animals without notochordal and with notochordal, and then of course the human. So we're particularly interested in whether you need to include pronase, what collagenase type and concentration that we're utilising, and what digestion times. Now, obviously, to test all of the different varieties of the methods that all the 36 groups submitted, that would take probably 10 years. So we have condensed it down into the most common methodologies that have been used and the rationale for those methods, and then utilising that knowledge to try and develop consensus methods testing within the laboratory. When we look at the NP cell expansion, we're particularly focused on the basal media and also the supplements which are being utilised. And what we're currently trying to look at is whether or not we can get rid of the need to use fetal calf serum in the media. The reason for this is, as I'm sure most people are aware, fetal calf serum is prone to massive batch variation. So if you're trying to develop a consensus methodology and every lab is still using fetal calf serum, it's not going to be the same, because actually that fetal calf serum is going to have very different concentrations of growth factors, of fibronectin, etc. So you're never going to get the same results in one lab to another if you've got different fetal calf serum. It's proving difficult to actually get rid of the fetal calf serum altogether during the cell expansion. So a key recommendation is that if you're working in consortia where you've got multiple groups trying to compare results directly, then you've got to batch test and all those groups utilise the same batch of fetal calf serum at least. It's a bit more promising when we go into the 3D culture systems, when we're looking at redifferentiation. And there it is looking like we can get rid of fetal calf serum. There's some alternatives that we're starting to look at. And we're particularly interested in maintaining the phenotype back to the in vivo situation, and of course the viability of the cells. Within these 3D cultures, we are doing some work looking at modifying oxygen and glucose concentrations, but that won't be fully explored within this first paper. The second paper that we've decided to pull out is that where we're looking at these large vacuolated cells, or what people term notochordal cells. And you'll see that I've kind of switched terminology throughout the talk. So these are the cells which are retained in those small animals, in pigs, and in some breeds of dogs. So these are the more juvenile cell types within the disc, which we need to actually look at these separately. They do have a different phenotype to the smaller, more mature NP cells. But when we were doing these discussions, it became very apparent that actually even just the nomenclature of these cells is very different depending on who you talk to. So this actually has stemmed a spin-off paper where we're trying to develop a perspective paper on what we actually call the cells of the disc, and what is the phenotype of these cells and how we can classify them. But once we've got what we're going to call them, basically it is those bigger cells that we're now looking at within this paper. And again, we want to develop a consensus methodology for how we can extract these cells, whether the species differences and what the digestion methods, etc., is that we're utilising, and then how can we maintain this phenotype in culture. These large vacuolated cells are well renowned to lose their phenotype in culture. They lose the presence of vacuoles, and they lose these more immature markers. There are a number of papers worldwide which have utilised different methods to try and maintain that, and those groups are all involved within the consensus paper. What we hope to demonstrate is how we can maintain phenotype and also give some recommendations of how people can characterise these cells. In addition, these cells are particularly hard to count accurately, and the problem there is because they come in clusters, it's very difficult to count cells when they're in clusters. So we've been developing methodology where you can actually pull those cells apart purely for the counting process, just in a small sample, so the rest of your cells you can still use as the clusters. Also, the freezing methods are not simple within these cell types, so we're also trying to make some recommendations for that within the paper. So the last thing I just want to show is basically all of the different people that are involved in this. It really is a fantastic large consortium that involves researchers all around the world. We've got representatives from every continent of the world, and lots of people are really engaged in this paper, well, in these papers. So I think the last thing just to say is thank you very much to all of the collaborators for all the work they're doing within their labs, and also the willingness to be involved in this exciting endeavour. And hopefully this will be a really excellent resource for spine researchers for years to come. So thanks for the opportunity to talk.
All right, thank you so much, Christine, for that. That was really interesting. I can get us started. There's a lot of variability between groups, and I was wondering, historically, how do you think that variability is developed? Do you think these groups, people came from different fields, came from different tissue types? Is there local variation from between universities?
Yeah, so this is actually one of the things we've been discussing, and we're going to put into the paper in terms of the reasons for this. It does appear that a lot of the variation is because of the very varied background of researchers, and often, they may be that they're the first person in their lab to use disc cells, so they use a method from a more similar cell, from the same lab. So it might look quite a lot of people have come from the chondrocyte field, and they use the methods that have been developed in the chondrocyte field across, which again, there's massive variation. But really, it actually just seems that methods are kind of passed from one person to the next, and you just use the method that's used in your lab, and then you take that method with you when you go and set up another lab, and it just passes along. So rather than actually, because there's so many papers using cells, no one actually knows which one to use, because every paper has a different method. And actually, if you look in the methodology of many papers, they don't have the level of detail of the cell culture requirements that people actually use as well. So, for example, if you take the pyruvate example, if you look in publications, hardly anybody comments whether or not they have pyruvate within their media. And actually that's an important energy supply, particularly if you've got a low glucose media. It probably doesn't matter too much if you've got the high glucose, but it's not included in the method. So how can people follow the method if it's not included?
Right. Actually, there's another question about sources of energy. So there's a question about glutamine. Is there data regarding the level of glutamine in the media?
So the trouble with glutamine is it breaks down very rapidly. So actually, a lot of people use the media which already comes with glutamine in, so the Glutamax, but then they actually add glutamine in addition. The concentration of glutamine doesn't really matter as long as you've got enough. So if you've got too much of it, it doesn't actually seem to matter for cells. But if you've not got enough, then obviously it does matter. We haven't actually collected concentrations of glutamine in people's media, mainly for the reason that it does break down really rapidly. So what you actually put in the start doesn't relate necessarily to what the cells are seeing. Mm-hmm. But you definitely need it in there.
Are there, here we go, another from James Artrides. So James says, outstanding talk, Christine, and excellent effort organizing this project. In terms of terminology for the project, I suggest you describe this as a systematic investigation, since a true consensus would involve additional steps, including more investigators beyond ORS spine section and specific voting on agreement, disagreement.
So actually, although I said that the initial sort of survey went out to the ORS spine section, it has also gone out to basically anyone that we knew that worked in the spine field. So we also sent it on to researchers who were not spine section members, but anybody. So actually anyone who's done cell culture has been invited to be involved. We are developing methods and testing it across different labs. So it will be recommendations. Obviously, we're not going to test it in all 36 labs. But I am hoping we will come to a consensus. We'll see with our fun Zoom meetings if we do. We might have to remove the word consensus if we have people who don't agree.
Christine, it's amazing work and so important, and I applaud you for that. In other disciplines, infection, for example, there's very rigid protocols on deciding on consensus. So we just, as we continue these discussions, we should explore that a little more. But I mean, I think for this group here, what you are onto the most important thing, which is answering these questions and bringing us all together more, which is really the key goal. Thanks.
Yeah, I mean, I really do think that in order for us to, progress in research, we need to talk to each other more. And I think that actually it's really nice that, nowadays there is there's much more sort of collegiate working, and, everyone is happy to work together. Whereas, a number of years ago, it was probably a bit more kind of competitive and things. But I actually think that now people are much friendlier and want to work together and things, which is absolutely fantastic. And that's how we're going to find a cure for low back pain, which really is what we want to do. So,
what was the total number of labs that you invited to participate, and how many responded?
So it's hard to say that because, of course, the ORS spine section email just went out to every member. So how many of those are actually cell culture users? So it's difficult to say exactly how many labs it went out to that were relevant. But in addition to the spine section itself, I think there were about 15 additional labs that were invited that weren't spine section members. And actually the majority of those people that were invited personally joined as well, which was great.
Okay. We got another question here from Hosni Sharif from Elizabeth Hagelin's lab. He says, nice talk, Christine. We know that MP tissue isolation is done with visual identification of the MP area. Is there any tips or consensus for the MP isolation to avoid mixture with inner AF cells? And also any validation steps like gene expression of MP markers to test, for example? Yeah,
it's a really good question, actually, because, you're absolutely right. The way we all isolate MP from AF is we look at it in the lab, and, when we're training our PhD students, it's basically, well, this is how you do it, and you give it a poke and you look for your striations, etc. The markers are very difficult because actually MP cells and AF cells, particularly those transitional cells in the middle, they express the same thing. So you can't really use gene expression and things. The only way I tend to do it myself personally when I'm training students in the lab is I'll always get them to, so when they've picked what's an MP and an AF, is they then have to embed it to wax, paraffin embed it, section it, and look at it histologically. And I'll only let them then do, the cell culture stuff once, yeah, that's definitely MP, that's AF, because really it is, it's once you look at it down a microscope. I mean, you can do things like, use dissecting microscopes and things like that. I don't tend to use that myself because actually I can just see it by eye. It's easy enough. But yeah, it is an issue, and whether you have got contamination from the inner AF and things in those samples. But I personally think they're a transitional cell anyway, so certainly in certain areas.
One last question, if you can answer it quickly. Kevin Burt from Nadine Shaheen's lab. Such a helpful talk, Christine. Will this study go into detailed comparison of how MP cells are isolated, similar to what you just said, dissection, separation from disc, pooling of multiple disc levels, in addition to growth and expansion media composition?
Yes, we hadn't actually included that in the chats, but yes, I think it is important. So yes, it will do now. So thanks for those comments. We'll include some pictures.
All right. Well, everyone, thank Christine for her hard work and for presenting that work to us. Thank you so much, Christine. And I'm going to turn it over to Sarah to introduce Sade.
Thanks, John, and thanks, Christine, for a great talk. Our next speaker is Sade Williams-Clayton from the University of Alabama at Birmingham. Sade completed her bachelor's degree in cell biology in 2011 at the University of Georgia. She performed undergraduate research in the labs of Dr. Michael Pierce and Dr. Boris Strypen. She joined the Graduate Biomedical Sciences PhD program in 2016 and is now a fifth-year PhD candidate in the lab of Dr. Rosa Serra. Her thesis work is focused on the development of the fibrous tissues in the spinal column, including the role of TGF-beta. Sade plans to defend her thesis in summer 2021 and is currently looking for postdoctoral positions. Sade, I'll turn it over to you.
All right, thank you so much, Sarah. Hey, thank you so much again for that introduction, and thank you so much for the organizers allowing me to present my research today. And today I'm going to be discussing how TGF-beta signaling mediates fibrous tissue differentiation during spinal column development, and a specific focus on the in vitro analyses and the techniques that I use in order to study this process. And I have no disclosures and I have no conflicts. And so like I said, I'm going to extensively talk about fibrous tissue differentiation, especially in the context of spinal column development. But let's first talk about why the spinal column is an important organ of study. And I know that for this specific group of individuals, I don't have to convince you that the spinal column is important to study, but I always start my talks this way. So let's dive right in. And so I first want to clarify what I mean by spinal column, because most people think I mean spinal cord when I say that, and that is very far from the truth. Again, what I mean are the bones and the discs of the spine, which is depicted here on the left. And we all know these stats, but let me repeat them for you. So it's important to study the spinal column because as we age or experience trauma, the IVD can undergo degeneration, contributing to chronic back pain. And we know that that is a leading cause of disability worldwide. It's an expensive disease that Americans and the world spends billions of dollars each year to treat. And again, it's an aging disease. And so by the age of 60, most of us on this Zoom call will experience some evidence of disc degeneration, which again can lead to chronic and severe back pain. Okay, let's delve into the IVD and those components. Again, I know I don't have to explain this to you guys, but I just want to make a point really quickly. And so again, the IVD is composed of the nucleus pulposus and the annulus fibrosus. And I'm particularly interested in the annulus, which is a fibrocartilaginous-like tissue, because again, as we age or experience trauma, this can degenerate, potentially leading to herniation, which can contribute to that severe chronic back pain that I mentioned. And so currently, there are no disease-modifying treatments. Currently, treatment for degeneration is treatment of the symptom, which is pain. And those treatments can be NSAIDs, epidural injections, maybe a little bit of PT to strengthen the musculature around the affected area. And the end-all, be-all, when all else fails is spinal fusion. Now, spinal fusion is a great treatment to treat pain by removing the affected area of the IVD that may be herniated or degenerated. However, fusion of those vertebrae can lead to accelerated degeneration in those adjacent discs surrounding that area. And therefore, there needs to the field is really lacking a therapy that targets the actual disease, which is degeneration, and not only the pain, but the pain is a significant thing that should be studied as well. And therefore enters my project and the goal of my project. And the goal of my project is honestly regeneration. My project aims to find the developmental pathways, signals, and cues that goes into the normal formation of the fibrous or fibrocartilaginous tissue of the annulus fibrosus, so that once we know those developmental cues, we can apply those cues later on in life for regeneration therapies. Now, our lab has made great strides in figuring out some of those cues that are involved in annulus fibrosis formation by discovering that TGF-beta signaling is required for the proper formation of the annulus fibrosis. And we did that by knocking out TGF-beta receptor 2 in collagen type 2 expressing cells, and by using a collagen type 2 Cree. And so what you can see here and appreciate in the control with this Austrian blue staining is that this lighter band of tissue here is the developing IVD. That circle in the middle is the developing nucleus pulposus, and all the tissue surrounding it in that lighter band is the developing annulus. With these larger, darker blocks of blue tissue are the developing vertebrae. So that's in the control of an E15.5 mouse, so early in development. Well, mid-stage of development. But when you knock out TGF-beta receptor 2 and collagen type 2 expressing cells, what you see is specifically that annulus is missing, again, indicating that TGF-beta signaling is required for the formation of the annulus fibrosis. So we know that TGF-beta is required for overall formation, but how? How is TGF-beta stimulating this development specifically of annulus, but not really affecting other pathways? Well, a recent paper that my lab just published, which came out in 2015, showed that TGF-beta acts as a master regulator, if you will, or a master switch. What happens is the components, those two major components of the spinal column, the the vertebrae and the annulus fibrosus actually come from a similar progenitor tissue called the sclerotome. And this sclerotome has the capacity to go down two different cell fates. It can either go down chondrogenesis to make cartilage, which again makes the bones of the spine, or it can go down the alternative cell fate of fibrogenesis, which makes the fibrous tissues of the spine, which include the fibrocartilaginous tissue of the annulus fibrosus, as I previously mentioned, or tendon and ligament. And what we discover is that TGF-beta acts as a master regulator by inhibiting and blocking chondrogenesis to allow the stimulation and acts as an instructive signal for these cells to go down fibrogenesis. And even before that, back in 2010, we actually solidified some pretty solid fibrous tissue differentiation markers. And these markers that I'm actually going to use for the rest of the talk to denote fibrous tissue differentiation are scleraxis, which is a transcription factor, and most notoriously known as a regulator of tendon formation, and fibromodulin, or FMOD, and atom TSO2, which are extracellular matrix proteins. So again, we knew the gross formation of the annulus fibrosus and that TGF-beta was required and a very important signal in this overall process, but how? This leads into my project. I really wanted to know how TGF-beta was mechanistically regulating this fibrous tissue development in the spinal column, and really solidify and find out those pathways that TGF-beta was utilizing to stimulate this response. So let's briefly go over TGF-beta signaling and the canonical and non-canonical pathways that's utilized by TGF-beta commonly in that progenitor cell population of the sclerotome. So TGF-beta acts as a homodimer that binds to a heterotetrameric complex on the cell membrane, and those consist of TGF-beta receptor 1s and TGF-beta receptor 2s. This stimulates a downstream cascade of activation of either canonical factors, which are SMADs, so SMADs 2 and 3, or it can stimulate non-canonical pathways that consist of AKT, P38, and as well as ERGs. And ERGs are in red here because, a quick hint, I'm going to really be focusing on ERGs for this talk. But TGF-beta can activate any of these pathways. And so I wanted to really figure out, was TGF-beta solely using non-canonical signaling, solely canonical signaling, or was there a mixture of all of these pathways utilized by TGF-beta to regulate fibrous tissue differentiation? Now, I want to briefly go over the methodology that I use in this study, and it is a solely in vitro study, so I wanted to show you what goes into that. Now, I isolate the sclerotome from E11.5 mice. Again, the sclerotome is that progenitor tissue that goes on to form the components of the spine. And this box is indicating essentially me isolating the spinal column. And when I zoom in, what you'll see is that this pink region in the middle is the developing spinal cord, and these bulbs coming off the side are the developing dorsal root ganglion. So the sclerotome is actually tightly adjacent to the developing dorsal root ganglion, and we can actually isolate them quite precisely because the sclerotome, for some reason, at E11.5 has a blue hue. So you can actually quite precisely isolate the sclerotome. Once I do that, I then plate these cells under micromass conditions. Now, micromass conditions is a high-density cell plating technique in order to mimic these, the condensed, very high condensation placement that the sclerotome is in naturally within the developing spinal column. So I plate these cells, which these balls indicate micromass conditions, because it's 3 times 10 to the 5 cells per 20 microliter of media. And so they're essentially in a little microenvironment by themselves. I leave that for an hour for them to become adherent to the plate. And then after they become adherent, I then treat them with TGF-beta for 2 or 8 hours. After that incubation time, I analyze fibrous tissue marker expression by Western blot and qPCR. Now, that's one version of this experiment that I do. The other is I isolate the sclerotome the same, but and plate them in micromass conditions. But first, what I'll do is then treat these micromasses with an inhibitor for different effectors, either in the canonical or non-canonical pathways. After 24 hours of incubation with those inhibitors, I then treat the cells with TGF-beta for 8 hours. And then after that, complete incubation time, again, analyze via Western or qPCR for fibrous tissue marker expression. Okay, so now since those methods are clarified, let's go into some data. So the first thing and all of this data is coming for my recent publication that just has been published. I got notification this Tuesday, so that's really exciting, in Scientific Reports. And so the first thing that we wanted to do was clarify that TGF-beta was regulating the fibrous tissue markers that we previously solidified in these cell types, which are the primary sclerotomal cells. And again, that's scleraxis, a transcription factor, and FMOD and ADAM-TSO2, extracellular matrix proteins. So we treated TGF-beta on these cells for 2 hours or 8 hours. And what we saw is that by 2 hours, only scleraxis was regulated by TGF-beta treatment. FMOD and ADAM-TSO2 were not regulated. Finally, at 8 hours, all 3 markers were regulated. And so it seems to be a difference in the temporal regulation of these markers, with scleraxis, that transcription factor, being regulated earlier. Now I wanted to tease out, did TGF-beta use any protein translation to regulate these markers? And this is kind of getting into, is there kind of an indirect or direct effect of TGF-beta regulating these markers? And the data that you're looking at here, by the way, is our RNA expression with qPCR data. So what we did to determine if there was protein translated needed to regulate these markers is treat these cells with cyclohexamide, which is an inhibitor of protein translation. What we see here is that, so this is control, this is treatment with TGF-beta, showing that scleraxis is regulated by TGF-beta treatment, just the control. But when we treat with cyclohexamide and cyclohexamide in the presence of TGF-beta, what we see is that TGF-beta still has the capacity to regulate scleraxis, even with protein translation being inhibited. And this kind of suggests to us that there may be a direct effect of regulation of TGF-beta on scleraxis expression. Now, what about those later expressed fibrous tissue markers? What we see is something a little different. Again, these first 2 bars are showing that TGF-beta can regulate these markers. Those are the controls. But what about, again, cyclohexamide treated versus cyclohexamide and TGF-beta together? What we see is that TGF-beta can no longer regulate these markers with protein translation inhibited, indicating that there needs to be probably some type of effector that TGF-beta needs to turn on that then helps regulate these markers. So this just shows, again, TGF-beta regulates these markers, but at different times and seemingly in different ways, with a special sort of regulation specifically on scleraxis. Okay, so that was establishment that these markers are being regulated by TGF-beta. Now let's get into the canonical signaling. Is TGF-beta using canonical factors to regulate these markers? So what we tested was was SMAD3 being utilized by TGF-beta to help with regulation. So what we did here, this is a Western blot, where again, we isolate primary sclerotomal cells and treat with TGF-beta for either 2, 8, or 24 hours. And the plus or minus is with and without TGF-beta. What we see is that over these different time points, TGF-beta does phosphorylate SMAD3 and does so significantly at 2 and 8 hours. Okay, now let's look specifically at the fibrous tissue markers. Does SMAD3 help TGF-beta regulate these markers? So the first thing we wanted to know, is SMAD3 required? Is it required by TGF-beta to regulate these markers? And to do that, we treated these primary sclerotomal cells with SIS3, which is the inhibitor of SMAD3 activation. And we tried it with 2 different concentrations. So what we did here is this black bar is treatment with TGF-beta alone. And the second 2 bars are the different concentrations of the SIS3, the SMAD3 inhibitor. What we see is that treatment with SMAD3 inhibitor and has no effect on scleraxis RNA expression, showing that TGF-beta doesn't need SMAD to regulate scleraxis. But these later expressed fibrous tissue markers of FMOD and ADAM-TSO2, once you treat with those increasing concentrations of SIS3, there's a downregulation in TGF-beta mediated expression of these markers, indicating that for FMOD and ADAM-TSO2, SMAD3 expression is required. But sclerosing, again, seems to be differently regulated by TGF-beta. So let's do the opposite experiment. Let's overexpress SMAD3 and see if that makes a difference. And we do this by adenoviruses. So the control adenovirus is GFP, and we overexpress SMAD3 with the SMAD3 adenovirus. And what we see is that if you compare the control virus to the SMAD3 virus, that SMAD3 alone, overexpression, is just not sufficient to regulate these markers by itself. These second two bars are the GFP virus and the SMAD3 virus, but in the presence of TGF-beta. And what this shows is that if you treat the virus, GFP virus with TGF-beta and SMAD3 with TGF-beta, still there's no significant difference between those viruses, meaning that simply SMAD3 is just not sufficient by itself to regulate these viruses. And sorry about that, PRG4 is actually a control for SMAD3 expression and is well known to be regulated by SMAD3. So essentially what this experiment told us that canonical expression yes, it is required for certain markers, such as FMOD and adenine-SO2, but not required for sclerosing, and it's not sufficient by itself. And even in the presence of TGF-beta, just isn't statistically different than the virus alone with TGF-beta. And so the question was, what else is happening? There has to be other effectors that helps to regulate the process of fibrous tissue differentiation. And so my hypothesis was that, TGF-beta is utilizing both canonical and non-canonical pathways to regulate fibrous tissue markers. And so just for the sake of time, we did try all of the, or at least most of the non-canonical markers to test whether TGF-beta was utilizing them to regulate downstream targets of sclerosing, FMOD, and adenine-SO2. We tried P38, we tried AKT, we tried NFAT, and we saw that, yes, that TGF-beta did regulate those markers, but those markers were not important, or those effectors, were not important in TGF-beta mediated fibrous tissue differentiation. But, hint, hint, ERK is, and that's why I'm showing this data. And all that other data is present in the paper that was just published. So what we did here to test if non-canonical factors were important, we did a similar experiment. We isolated those primary cells, treated them with TGF-beta for 2, 8, and 24 hours. And what we saw is that ERK is significantly regulated by TGF-beta at each time point, as you can see here denoted in this graph. And so what we wanted to do now is inhibit ERK activation to see if that had an effect on these fibrous tissue marker differentiation. So this is showing us inhibiting ERK by using a MEK inhibitor. And so MEK is the kinase directly upstream of ERK that activates ERK by phosphorylation. And so what we did was use 3 different concentrations of that inhibitor in the presence of TGF-beta and saw that that inhibitor blocked TGF-beta mediated activation of ERK. And that is shown in the graph here, and that's all significant. Now let's look at fibrous tissue expression. What we saw is that these are the 3 fibrous tissue markers. The black bar is TGF-beta alone, and these 3 other bars are the increasing concentrations of the ERK or MEK inhibitor. And what we see is that without ERK activation, TGF-beta cannot regulate any of the 3 markers, which is extremely interesting because, again, sclerosing always seem to be differentially regulated by TGF-beta, and it seems to be regulated specifically by ERK, as well as the other later expressed fibrous tissue markers. Now, we briefly wanted to explore, can TGF-beta also regulate sclerosing to help regulate later expressed fibrous tissue markers, aka FMOD and adenine-SO2, since it is a transcription factor and it is regulated slightly different than the other markers. I tested this hypothesis in C3H10T1/2 cells, which is an immortalized mesenchymal stem cell that mimics primary sclerotomal cells. I knocked down sclerosing expression. This is a Western using siRNA, and you see that it is significantly knocked down. And then I looked at RNA. Of course, sclerosing is knocked down with the sclerosing siRNA versus scrambled. And then I looked at FMOD and adenine-SO2. And what it shows here is that these first 2 gray bars are the control siRNA. These last 2 are sclerosing siRNA. And that if you look at the control siRNA and the sclerosing siRNA without TGF-beta, then basal expression of FMOD is downregulated. And if you look at just sclerosing siRNA, but without and with TGF-beta, what you'll see is for both FMOT and ATOM, there's no significant difference. And what that means is that if sclerosing was not needed by TGF-beta to regulate these markers, then treatment with TGF-beta would have been a large upregulation in this marker's expression, as we see here in the control, which is control siRNA treated with TGF-beta. But there's no difference, meaning that TGF-beta cannot regulate these markers without some help from sclerosing being present. Since this is a knockdown, so it might be like without enough concentration of sclerosing present, TGF-beta cannot regulate FMOT or ATOM TSO2. So that was really interesting because there seems to be this convoluted pathway of how TGF-beta are regulating these fibrous tissue markers to stimulate fibrous tissue differentiation. And I want to try to bring all that into summation with this visual model, where in the primary sclerotome, we zoomed into one of those cells, you will see that TGF-beta causes a downstream pathway where ERK is regulated very early, at least by 2 hours of treatment, probably very much earlier, and stimulates the expression of sclerosing RNA, which this protein of sclerosing in combination with SMAD3, we don't know if it's direct or just a cooperation, goes to then turn on these later expressed fibrous tissue markers, which in summations stimulates fibrous tissue differentiation. Now that leads me to some conclusions and implications of this data. So it supports a novel pathway used by TGF-beta, but it does leave us with a few questions. How is TGF-beta regulating sclerosing? Does sclerosing and SMAD3 interact directly, or is it just a cooperation? And how is ERK, which is a kinase and not a transcription factor, specifically regulating these fibrous tissue markers? And again, this study can be used in regenerative therapies to treat degeneration, probably in a combination of stem cell therapy and tissue engineering strategy to address degeneration in these fibrous tissues.
Thank you so much, Shade, for a really great talk and a lot of comprehensive studies.
First, I would like to thank Sarah and John for organizing this paper, how you call it, journal club. And Sid and Christina for the nice presentation. I have a question with Sid. Did you look at the TGF-beta receptor that are on the cell? Because we just published, Martin Stoddard, in fact, from ARI, that the ratio between type 1 and type 2 receptor, TGF-beta, it's important for chondrogenesis or non-chondrogenesis. So that's the question.
So no, we did not explicitly look at the concentration or the localization of the TGF-beta receptor because we know from the literature that it's ubiquitously expressed, and if it's there will be signaling. So we didn't really focus on the concentration.
Because depending on which receptor it binds, it could have a different intracellular effect, say like that.
That is very true. So depending on if it's, especially for receptor 1, it's extremely important what type of receptor, whether it's ALK4, 5, or 7. Yes, what's present, but we didn't focus on that because we saw essentially an effect. We didn't focus on or try to tease out which receptor was causing or activating which effector. But that is something that we could go back and focus on to really pinpoint more specifically what exactly is happening.
Thank you.
You're welcome.
Great, Sade, excellent talk. And I missed some of the beginning, but it's such interesting questions and really clearly presented. It's really wonderful work, and also congratulations to the organizers for a really fabulous session. Question, Sade, can you help us understand, right, the sclerosing knockout, the early paper from Ronan Schweitzer and Price, it didn't seem to have a disc phenotype. So can you help us to understand, and we've been using it as a marker and thinking that it probably has functional importance, and not just for identifying cells, but also as a potential phenotypic marker and not just a marker. So can you help us understand how your results help us interpret the global knockout study? Were they just not looking at it right, at the disc specifically enough to see more subtle changes, or is it a complex interaction?
Of course, they were looking at it right. Ronan is a fantastic scientist.
Oh, yeah, and I don't mean that. I mean, of course, they do excellent work, but maybe they weren't looking precisely as a disc-specific worker might.
So I'm glad that you asked that question, because that's one of the main questions that I get, right? The knockout doesn't have a phenotype. So what's the point? And the point is that I wish they did a more, as you mentioned, comprehensive look at the discs. So they looked at one specific time point. I think it was E13 or E13.5, which around that time, the structure and the organization of the disc should be solidified. But you really, again, these are just a glance at one specific embryonic day. And I wish they would have done a timeline, because their focus was the tendon. And I understand, resources are limited, but I wish that paper really focused on the disc through different embryonic days. How did the collagen organization look? Were there the biomarkers that are usually expressed in the disc? Were they normally expressed there? How did that phenotype hold out? Well, going towards later developmental days and even, around right after birth, how did it look then? And so I wish there just would have been a fleshing out of focusing on the disc phenotype. And also, I believe this is an extremely complex network of transcription factors and other factors that goes into this formation. Because the disc itself is such a complex structure where these, there is an inner and an outer annulus and also a nucleus pulposus that shares some of the molecular markers with those two structures. And I think that why we specifically focus so, keenly on these specific markers is because it's so complex, and we really needed to break it down to, specific targets so we can figure out a pathway. Well, now, okay, we figured out a central pathway. What about all the other markers that we found for the disc? Do they fit into this pathway? What are the web of, pathways and interactions that are involved? So I think that though, that paper showed no phenotype, I think there's more that meets the eye, because these are such complex pathways, and the timing is also so important as well. So I think that it's just more that needs to be studied, in short.
That seminal work needs more work. Fabulous. Great answer. Thanks, everyone.
Okay, thank you so much, Shade. I really appreciate it. And thank you, Christine, for a great talk as well. We're all out of time for today, unfortunately, but thank you all for joining us, and let's thank our speakers again. And this concludes our virtual scientific session.